Good evening, everyone. Look, I might just start slowly. We do have to run these webinars to a pretty rigid timetable. The purpose of these webinars is to facilitate exchange around the Adelaide Biomed City Precinct, facilitating collaborations and for our own scientific interest. You may be pleased to know that the previously recorded webinars should be ready within the next few days or so, and tonight's session is also being recorded. So for those that you've missed earlier ones, you'll be able to catch up. Tonight we have Professor Michael Brown and Associate Professor David Ross. First of all, Professor Michael Brown is going to talk to us about the Royal Adelaide Hospital Cancer Clinical Trials Unit and opportunities for the precinct. So I'll ask you to start your presentation, Michael. There we go.
So I hope that is displaying.
Yep.
So thank you very much for the opportunity to speak. Some disclosures related really to interactions with the pharmaceutical industry. I'll talk about the activities of the Cancer Clinical Trials Unit first. So it's a manager with 12 coordinator staff. It's primarily associated with the Royal Adelaide Hospital Medical Oncology Department, which has 10 consultant staff operating across all tumour subtypes. And we have a portfolio at any one time of over 40 active trials, which range from phase 1 through to phase 4 or post-marketing trials. And these are of all tumour subtypes. Most of the trials are sponsored by the pharmaceutical industry, but others are also funded by cooperative study groups, mainly NHMRC sponsored, and then there are some investigator-initiated trials which are often sponsored by CAHLN or Central Adelaide Local Health Network. My particular interest and expertise and experience is in first-in-human and phase 1 clinical trials, particularly of cancer immunotherapies and including cell and gene therapies. And it's worthy to note that we've embarked upon a national collaboration of all states and territories, where Royal Adelaide is the South Australian node of something called the Australian Genomic Cancer Medicine Centre, federally funded program, and it allows genomic testing of patients' tumours to match targeted treatments, usually on clinical trials. One of those trials is the MOST trial, and there are also surveillance and genetic testing programs for young adults, that is 40 and under, who have been diagnosed with cancer. In addition, I have a lab which is based at the Centre for Cancer Biology in the UniSA Health Building, adjacent to the Morford Street Bridge. Our main objective is to translate from bench to bedside and occasionally from bedside to bench phenomena related to cancer immunotherapy, but also development of new products, and that's what I will turn to in a little bit. One of the Cancer Australia funded projects we have is to look at immune signatures in blood that would tell us before patients start the most common form of cancer immunotherapy, immune checkpoint inhibitor therapy, whether it can work. And we've discovered this in melanoma and looking to apply it to other cancers. We also have an active chimeric antigen receptor CAR T-cell therapy program, which is both clinical and preclinical. There is an ongoing phase 1 clinical trial, originally in melanoma patients, now extending to patients with other GD2 positive malignancies. And then we're looking at brain tumours in collaboration with a group in Sydney, and also a trial based at Royal Adelaide Hospital upcoming in the next year for both. The preclinical work is done in this laboratory, has a focus on novel targets in glioblastoma, but also looking at new tumour homing strategies, because this is one of the problems with targeting brain tumours. And the key researchers are listed there. There's another technology that's also an immune-targeted technology based on antibodies, and this is trademarked Apomab, owned by OzHealth Proprietary Limited on behalf of CAHLN, and it's for detecting and predicting tumour responses to DNA-damaging anti-cancer treatment arising from dead cancer cells that have been killed as a result of that anti-cancer treatment. There is a immuno-PET phase 1 clinical trial currently open, and the second patient is enrolled now. And there is a program of work that is preclinical related to this, and the key researchers are named there. There are various things as a clinical trials unit that we can offer, as many of them do in the hospital: access to patients' biosamples, a mechanism for running trials through a scientific review with investigational drug subcommittee, and then there's also capacity within the hospital to have overnight stay through a unit called Park Research. We interact with many global and pharmaceutical and biotech companies, as well as cooperative study groups, other cancer institutes interstate, and particularly Baylor College of Medicine, Houston, for the CAR T-cell therapy. Just to give you an example, which was part of the brief, of what we've done in a multidisciplinary collaboration. This antibody I've talked about over about 10 years now was first of all cloned and developed at CSIRO in Melbourne, and then the Australian Institute of Biotechnology and Nanotechnology made a producer cell line for the antibody. The preclinical studies were done in my lab, and then SAMRI has been very helpful in collaboration to make the radio label, zirconium-89, and its use its radiopharmacy. Our trials unit has put this through the drug subcommittee and the Human Research Ethics Committee, and the patients are being scanned at the Clinical Research Imaging Centre, a joint enterprise between SAMRI and Dr. Jones and partners. This has led to pharmaceutical company interest, and there's been a licensing and option agreement executed between OzHealth and that company. We have some gaps still we would like to fill. One of them is a secure CAR T-cell manufacturing facility. I think biostatistical support is lacking generally in this precinct, and structural biology doesn't exist at all in South Australia on any scale. There's a new institute to start. It's already been established as a partnership between University of Adelaide and Central Adelaide Local Health Network, so more of that later. Free to offer consultancy services, and I can be contacted there. So thank you.
Thanks very much, Michael, for that excellent presentation, keeping to time. There is a Q&A function that I would invite the participants to use, and I'll monitor the questions. We've got 5 minutes for questions. I thought I might just ask Michael, the new CAHLN facility, is that going to fill some of the gaps that you identified towards the end there, do you think?
Well, probably not biostatistics. But certainly we aim to develop an infrastructure for CAR T-cell therapy, and structural biology is also our aims, one of our aims. People may or may not be aware though, the kit, the equipment for structural biology is enormously expensive and not really part of the funding package, so that can come to tens of millions of dollars. So it probably requires more general interest than outside of this institute for it to be viable.
Okay. And the clinical grade production of the APOMAD reagents, is that done locally?
Well, no, it's not clinical grade. It's been GMP-like great. And that's permitted under a clinical trial notification, which is how this has gone forward with TGA approval. And also the radiopharmaceutical arrangements at SAMRI under a TGA exemption.
Right.
That's one of the advantages of our regulatory system for getting early phase studies done.
Right. And you won't have any problem upscaling that for the number of patients you hope to enrol?
Not for what we plan to do, because the dose is tiny. So the dose of protein is only 1 to 2 milligrams, and we got grams.
Okay, excellent. Janelle's asked a question. What is the status of cell-free DNA testing for cancer?
In blood, you mean? Or
I'm guessing that's what he means, circulating.
Well, we don't have any tests that are routine. They're research-based tests. They don't have any, there's no approved tests that I'm aware of. Part of the problem is sensitivity, that people can have tumours without, with the mutation present, but not detectable in blood. And one major company attempted to get such a test approved, but not really, this is actually for lung cancer, your area, but not able to proceed because they didn't have sufficient data to support using it versus looking in the tumour itself. So I think there's quite a way to go.
Sure. And just from a local, pragmatic, practical point of view, has the park facility been upset with COVID shifting, and are you good to continue with phase 1 studies at the moment?
Oh, it's had to shift its operations within the hospital because of COVID demands. So it's been relocated, but operational in the hospital.
Okay, that's good to know. Any other questions on the Q&A function? I've only just got that one, and hope I'm not missing any. So with the APOMAB, which has obviously been your, main area of development, I think you said, is it just the 2 patients that you've had?
Yeah, that have been, well, we've only just started in the last couple of months to be able to recruit. But these are patients who do not have current treatment for their cancer, so either any kind of lung cancer or ovarian cancer, who are being observed and not undergoing active treatment. So we have 2 of 6 we need to recruit in that category. Once that's complete, then we can turn to recruiting patients who will get the APOMAB after an injection of chemotherapy, actually chemoimmunotherapy.
So these patients are naive, they've not had any prior chemotherapy?
No, they may have had treatment, it's just they're not being actively treated. So the point of doing this was not to have in the first injections the confounding factor of adverse events related to chemotherapy.
I see. And so this is purely a safety study because you've not killed the cells to allow the antibody to bind to the dead cell?
Right, for the first 6 patients. Yep. So just to show you, it's a chimeric antibody, so it's got mouse antibody components. Very unlikely people would react. Right. And it's never been administered to humans, so this is the reason for this extra degree of caution.
All right, well, look, thanks very much, Michael. As I said, it's a tight schedule, and we'll wrap it up there. And we'll hand over to Associate Professor David Ross, who's now going to talk to us about the South Australian Cancer Research Biobank. So, David, if you wouldn't mind sharing your screen and giving us your talk.
Right, thank you for the invitation to speak. So this is a little bit of a change of tack because we don't actually do any research ourselves in the Cancer Research Biobank. We're an infrastructure project that enables other people to do research. So I'm a clinical and laboratory hematologist in the Royal Adelaide Flinders Medical Centre and SA Pathology, and I have research appointments in SAMRI and the CCB. So I have many links across the precinct. I've only relatively recently taken over as director of the biobank following Victo's retirement. So this has been his baby for the last 20 years, really. So the Cancer Research Biobank is a project that exists to enable local cancer researchers and potentially their national or international collaborators to undertake projects that require potentially large numbers of clinically annotated patient samples. So I guess the principle of this is if you came along saying you wanted 50 samples from people with lymphoma, if you started the project today, it might be a year or two before you could do your work. But if we've got freezers full, you can get going straight away. So it really is an important enabling project for lots of different research. So since this began as a hematology bank, although we are open to banking other types of cancer if people are interested in sharing our infrastructure, we primarily collect blood and marrow samples at the time of a bone marrow biopsy. We store viable cells and also plasma, serum, and on request other materials such as hair follicles or buccal swabs for germline testing. We also have permission to store leftover diagnostic samples. So if there's DNA or RNA left over after diagnostic testing, that can also be donated to the SACRB collection for future use. In addition, we collect normal bone marrow from young, healthy donors. So they're mostly penniless university students who are happy to donate some marrow for a small honorarium. And that provides a source of cells for normal controls for a range of experiments. And perhaps importantly, in addition to the archive of samples that we have, we provide an ethics and governance framework. So essentially, once the samples are in our collection, they're covered by our ethics approvals for future unspecified research, which means that if samples are collected for a specific purpose, they're locked into that purpose, whereas our approval is for future unspecified research, so long as that research has ethics approval. So this creates a degree of flexibility that in itself is very useful. Importantly, essentially the ownership of the samples sits with a committee of users. So the clinicians who have an interest in myeloma and the researchers who have an interest in myeloma together form a committee that governs the release. So there's ownership from the involved parties to control the use of those samples. Because of the time constraints, I've just got a couple of examples of the kind of research that can be done. So these are both quite small projects. So this is one of the myeloma projects from Andrew Zanetino's laboratory. So they were interested in looking at genetic factors that influence the transition from monoclonal gammopathy, which is a pre-malignant condition, to smoldering myeloma, which is a clinically asymptomatic condition, through to overt myeloma, which is a life-threatening malignancy. And so they were able to get longitudinal samples where a patient has had bone marrow biopsies many years apart and track the changes in the genetic makeup of the clone. Importantly, this work was undertaken with collaborators from overseas, including Gareth Morgan, who's from Wales, and Charles Mulligan, who's from Adelaide, but is now in St. Jude in the US. And here's an example from one patient. So you can see that over samples taken more than 5 years apart, you can see that most of the clones are stable, whereas some have expanded slightly and some have contracted. But remarkably, considering the dramatic clinical change, there's not a lot of change in the clinical structure. So you can imagine this kind of work is really only possible because we've got an archive that goes back for 10 or 20 years. And that means that we can provide samples spaced a long way apart that represent the whole of the patient's journey. In a different case, so this is one of my own patients. This example is a single patient who had a previously unrecognized PDGFRB translocation. So PDGFRB translocations are a CML-like illness that commonly causes eosinophilia. This particular PDGFRB rearrangement is cytogenetically cryptic because it's a small interstitial deletion. So we were able to find this previously unrecognized deletion, which was sensitive to imatinib. And by tracking back to past samples and to forward to future samples during treatment, we were able to show that there was originally a pre-leukemic clone, that the PDGFRB translocation was acquired, which caused expansion. Additional lesions occurred with disease progression. And then you can see that the majority of these advanced lesions disappear with imatinib treatment. And of course, this was clinically important because it enabled this patient to receive a therapy that would not otherwise have been available. So going back to a little bit more of the mechanics of the bank. So we're funded by the University of South Australia and the University of Adelaide. SAMRI doesn't provide us with direct funding, but accommodates us. So the bank is located in the basement of SAMRI. So there's a substantial in-kind contribution. And in addition, we're funded by by CAHLN, SA Pathology, and the Health Services Charitable Gifts Board. And it means that the institutions that fund us then have essentially free access to the samples. So when people from those universities or from SAMRI are requesting samples from us, there's generally no charge because of the infrastructure contribution that those institutions have made. Since we don't do any research ourselves as a biobank, the outcomes of our work are measured in the output of the researchers that use our samples. So here's a very brief summary from the last 5 years. So you can see that most years, grants using samples that we have collected bring in about $5 million to the precinct. And we typically have about 15 to 20 publications arising from the use of those samples over the course of the year. So although it's a substantial ongoing commitment to keep the bank going, the work that it enables is also very valuable and really pays for itself. In addition, we think it's important to create research opportunities to help with the recruitment and retention of staff who have academic interests. It creates opportunities for students and researchers. And I think it makes us attractive to international collaborators because the bank has been in existence now for more than 20 years. We have quite large collections in many diseases, and that makes us an attractive collaborator for some big international groups who are looking for large numbers of samples. In addition, it means that we can rapidly translate research findings to clinical care, and some good examples of that come from the discovery of germline mutations that cause predisposition to cancer, like GATA2 and DDX41, for which some of the original publications came from South Australia, from SACRB. We hold around 150,000 samples from more than 5,000 individual patients. We typically have about 10,000 new samples in per year, and we release about 1,500 samples to researchers. Of course, this means that our collection is growing every year. It constantly gets bigger, so we're keen to increase our output. And so if there are other researchers in South Australia who think that they can make use of this resource, we'd be very happy to hear from those people, because really our productivity is our output, not the things that we have sitting in the freezer. Finally, I'd like to acknowledge all of the funding partners whom I've mentioned during the talk, and particularly Professor Toe, who established the South Australian Cancer Research Biobank, and to our staff and to the many patients who provide us with our samples. Thank you very much.
Thanks very much, David. One question that comes to my mind is obviously this is predominantly a haematology-based bank, or has grown that way. If hypothetically I was wanting to deposit some lung cancer samples there, how accessible is this? Was there a mechanism for starting up new diseases? How does it work?
Yeah, so we've had discussions with some other groups who have archives of samples that they'd like us to help with. So we would be open to that. So because we work with these disease committees who control the use of the samples, we would expect that if you had a group of researchers and clinicians, that you would form a committee to control your samples. They would have to be either linked through our database so that you could physically keep them and essentially share our ethics processes. So the main thing would be the ethical approval that your samples can be treated the same as the rest of our samples, but where they physically sit would be negotiable. Of course, because of the costs associated with actually moving samples into our bank, if that were the case, then we would obviously need to discuss some kind of funding support to increase the scale of our bank. But obviously, the more that we hold, the cheaper it is per sample because of the fixed sort of background costs that go into running things.
I see. There's a question from Kevin Patterson. Do you envisage collecting samples via opt-out consent forms? Any facility to collect longitudinal clinical data?
So we haven't permission to have opt-out. So because of the potential uses, including genetic testing, the ethics committee has been reluctant to allow an opt-out. So I think it probably wouldn't meet the NHMRC requirements for waiver of consent. So I don't think that that is possible for the bulk of our collection. And sorry, the second part of the question?
Any facility to collect longitudinal clinical data?
Yes, yeah. So we do. So in the course of our, many patients will have a bone marrow biopsy at diagnosis and then at relapse and to assess response to treatment. So because of the way that our bank has been structured, we naturally collect longitudinal samples. In addition to which, if somebody wanted to contribute a blood sample or some other sample along the way without a marrow, that's also possible. So I think that's one of the key strengths of our bank, and from the examples that I showed, is that we do collect longitudinal samples. So for some of our patients, we would have 5 or 6 samples from before and after different rounds of treatment. And that enables us to study factors that are predictive of response to treatment and factors associated with relapse and progression.
Cadence Haynes has asked, what is the process for new research groups to access samples from the biobank?
So we have an application process. So if people approach us, we can send the application form. So essentially, the researchers need to tell us what they want to do with the sample and provide us with a copy of ethics approval. And if they're affiliated with one of our funding institutions, we would then, if the project is approved and we have appropriate samples, we will then release them to those researchers. So we've had a couple of groups approach us for the first time this year, and that's the process that they've followed.
And I think you indicated if they are affiliated, the costs are negligible, is that?
Yes, so essentially the annual contribution that comes from the universities would cover releases to those universities. If people had special requirements for data collection or processing, then there might be an additional charge for that. But for the release of the number of samples that's required, there's generally no charge.
Melissa Bennett's asked, does the general ethics have specific approval for animal work?
So the ethics committee required an opt-out for animal studies. So there's an opt-out. So most of our recent samples would have a clear instruction as to whether or not the patient had consented to xenografting or other animal experiments.
I see. And in terms of the logistics of maintaining this into the future, you've obviously got a lot of samples already. What's the capacity? When will you be maxed out?
We've recently, we're actually in the process of relocating into Samri, but we've got new tanks, so we've got capacity for years to come. I don't know when we'll reach saturation. I guess that depends on how many we send out. It's actually not economical to go through and weed out your collection and discard things. It's cheaper to buy a new tank and start filling that than it is to go weeding out old samples. So I think as long as we remain productive and the institutions continue to fund us, we'll just keep storing samples. And essentially, there's no maximum number at which you don't need to collect, because as the treatment landscape changes, new targeted therapies come in, you want samples from people who've seen those new drugs, which means that you can't just rely on things that you stored 10 or 20 years ago. So we have to constantly collect so that we're constantly reflecting current treatment landscapes.
And do you have like standard operating procedures for how the samples are collected? So fresh, formalin, or how does this work?
So essentially the samples are sent to us from routine diagnostic procedures. So they're collected as part of normal clinical care. We have standard operating procedures within the bank for how we process those samples, but we don't control how the samples got to us because they're generally collected as either as part of a clinical trial or as part of a routine diagnostic procedure.
Right. Well, look, it looks like we're out of time, but that sounds like obviously it's a fabulous resource, and it's been extraordinarily productive, and perhaps it's stimulated some people out there to contact you with some ideas, maybe either using what you've got or possibly even expanding it slightly. But we are now out of time. As I mentioned at the beginning, this, along with previous presentations, have been recorded and will be available, I'm told, in a few days' time. I'd encourage you to keep this time slot in your calendars for next week. And I'd like again to thank both of our speakers, Michael Brown and David Ross, for their very interesting presentations. And we'll close it there. Thank you.